Good morning, students. Our today's topic is meiosis. The production of offspring by sexual reproduction includes the fusion of two gametes, each with a complete haploid set of chromosomes. During meiosis, reductional division occurs, haploid gametes are formed, and after fusion, during sexual reproduction, again diploid number of chromosomes are maintained. Gametes are formed from specialized diploid cells. This specialized kind of cells division that reduces the chromosome number by half results in the production of haploid daughter cells. This kind of division is called meiosis. So in meiosis, reduction is also known as reductional division, in which the number of chromosomes are reduced to half. Meiosis ensures the production of haploid phase in the life cycle of sexually reproducing organisms, whereas fertilization restores the diploid phase. In sexually reproducing organisms, meiosis represents the haploid phase of the life cycle. And after fertilization, the diploid phase again is maintained. We come across meiosis during gametogenesis in plants and animals. This leads to the formation of haploid gametes. The key features of meiosis are as follows. Meiosis involves two sequential cycles of nuclear and cell division called meiosis 1 and meiosis 2, but only a single cycle of DNA replication. So this is the first difference between meiosis and mitosis. In mitosis, only mitosis one phase occurs. Mitosis only is there, but in meiosis it occurs under two cycles. First is meiosis 1 and then meiosis 2. Meiosis 1 is initiated after the parental chromosome have replicated to produce identical sister chromatids at the S phase. So meiosis 1 starts after the replication of the chromosomes. Meiosis involves pairing of homologous chromosomes. What are homologous chromosomes? Each chromosome is in a diploid organism. One chromosome is contributed by the mother and one chromosome is contributed by the father. So for one character, two or one pair of chromosome is there, that is known as homologous chromosome. And recombinations between non-sister chromatids of homologous chromosomes. So what are non-sister chromatids? So if these are the two arms known as chromatids, they are joined in the middle by the centromere. The two arms which are joined with the same centromere are known as sister chromatids. And with the other homologous chromosome, the arms from the other homologous chromosome is known as non-sister chromatids. 4 haploid cells are formed at the end of meiosis 2. In mitosis, 2 cells are formed, but in meiosis, 4 cells are formed after meiosis 2. Meiotic events can be grouped under the following phases like Prophase 1, Metaphase 1, Anaphase 1, Telophase 1. Similarly, Prophase 2, Metaphase 2, Anaphase 2 and Telophase 2. So first, meiosis 1. Under meiosis 1, first phase is prophase 1. Prophase is the first meiotic division, is typically longer and more complex when compared to prophase of mitosis. In meiosis 1, prophase is more complex as compared to the mitosis. It has been further subdivided into following 5 phases based on chromosome behavior. They are lactotene, zygotene, pachytene, diplotin, and diakinesis. So first learn these 5 names in sequence only: leptotin, zygotin, pachytin, diplotin, and diakinesis. During leptotin, the compactation of chromosomes continues. This is followed by the second stage of prophase 1 called zygotin. So mainly in leptotin, the chromatin material changes into chromosomes. Loss of water occurs from the chromatin material, or they take a proper shape and becomes chromosomes. Compactation of chromosomes occur. Next is zygotin. During this stage, chromosomes start pairing together, and this process of association is called synapses. Main point is synapses. During zygotin, synapses occur. Such paired chromosomes are called homologous chromosomes. During zygotin, the homologous chromosomes come close to each other, and this process is known as synapses. Electron micrograph of this stage indicates that chromosome synapses is accompanied by the formation of a complex structure known as synaptonemal complex. When the homologous chromosomes come close to each other, this process is known as synapses. The whole complex structure is known as synaptonemal complex. The complex formed by a pair of synapsed homologous chromosome is called bivalent or tetrad. However, these are more clearly visible at next stage. The first 2 stages of prophase 1 are relatively short-lived compared to the next stage, that is pachytin. So first, in leptotin, compactation of chromosomes occur. During zygotin, synapses of chromosomes occur, that is homologous chromosomes come close to each other. Third is pachytin. During this stage, the 4 chromatids of each bivalent chromosomes become distinct and clearly appears as tetrads. Now all the 4 chromatids are clearly visible in pachytin. This stage is characterized by the appearance of recombination nodules, the sites at which crossing over occurs between non-sister chromatids of homologous chromosome. So main point in pachytin is crossing over occurs between non-sister chromatids of homologous chromosomes. When the homologous chromosomes come close to each other, the crossing over occurs between the non-sister chromatids, which creates new combination in the presence of an enzyme recombinase. Crossing over leads to recombination of genetic material on the 2 chromosomes. Because of this crossing over, new combinations appear. Recombination between homologous chromosomes is completed by the end of pachytin, leaving the chromosomes linked at the site of crossing over. So in pachytin, crossings over occurs, new recombinations appear. Next is diplotin. Diplotin is recognized by the dissolution of the synaptonemal complex and the tendency of the recombined homologous chromosomes of the bivalent to separate from each other except at the site of crossover. During diplotin stage, synaptonemal complex dissolves, and that recombinase complex, the homologous chromosomes which have come close to each other, now they also start separating except at the X point, X-shaped structure. At one point they have crossed their arms. Crossing over occurs. That X-shaped structure is known as chiasmata. So learn the word chiasmata. It occurs in the stage diplotin. The X-shaped structure called chiasmata, that is a crossing between the chromatids of a homologous chromosome, non-sister chromatids of homologous chromosome. This X-shaped chiasmata is present. In oocytes of some vertebrates, diplotin can last for months or years. The final stage of meiosis, prophase 1, is diakinesis. This is the fifth stage of meiosis 1. This is marked by terminalization of chiasmata. Now this end of the chiasmata occurs. During this phase, the chromosomes are fully condensed and the meiotic spindle is assembled to prepare the homologous chromosomes for separation. By the end of diakinesis, the nucleolus disappears and the nuclear membrane also breaks down. So same features as in the end of the prophase of the mitosis we have studied. Nuclear membrane disappears, nucleolus disappears. So diakinesis represents transition to the metaphase. After diakinesis, metaphase occurs. So in meiosis 1, first learn the 5 names. Leptotin. In leptotin, compactation of main feature is compactation of chromosome occur. In zygote, synapses or synaptonymal complex is formed. In pachytene, crossing over occurs. In diplotin, X-shaped structures called chiasmata is formed. And in diakinesis, end of the chiasmata or the formation of spindle starts appearing. Nucleolus disappears, nuclear membrane also disappears. Next is metaphase 1. The bivalent chromosomes align on the equatorial plate, same as we have studied in mitosis. The microtubules from the opposite poles of the spindle attach to the kinetochore of the homologous chromosome. Here is the difference from the mitosis. In mitosis, from the pole through spindle fibers, the chromatids are attached. The centromere splits in the mitosis. But here in meiosis, the complete chromosome goes to the one pole. So here the one homologous chromosome, out of homologous chromosome, one chromosome is linked to the one pole and other chromosome is linked to the other pole. So the microtubules from the opposite pole of the spindle attached to the kinetochore of the homologous chromosomes. Next is anaphase 1. The homologous chromosomes separate while sister chromatids remain associated at their centromere. Here centromere do not split like in mitosis. Here the one complete chromosome goes to one pole. Out of the homologous chromosomes, one chromosome go to the one pole and other chromosome go to the other pole. So here the actual reduction of chromosome occurs during anaphase 1. Telophase 1. The nuclear membrane and nucleolus reappears. Cytokinesis follows and this is called the dyad of cells. Now from one cell, two cells are formed. Although in many cases the chromosomes do undergo some dispersion, they do not reach the extremely extended state of the interphase nucleus. The stage between the two meiotic division is called interkinesis. The resting stage between the meiosis 1 and meiosis 2 is known as interkinesis and is generally short-lived. There is no replication of DNA during interkinesis because here in meiosis replication occurs only once, that is before meiosis 1. Interkinesis is followed by prophase 2, a much simpler prophase than prophase 1. So after telophase 1, the cell goes into the meiosis 2 condition. In that again prophase 2, metaphase 2, anaphase 2 and telophase 2. That we will study next time. Read this one. If you have any doubt, you can ask. Thank you.